Thank you, Matheus. So thank you once more, Mariana, to invite me. I'm always glad to be invited by Mariana. It's always a pleasure to be here. I was a student from here. Mariana always remember me, so I'm very happy. And Wagner also. Where is Wagner? Oh, here he is. So thanks a lot. Um, for today, I will talk a little bit different of the other talks that have been done in the last months because Mariana just asked me to focus in mastoparans. I'm not seeing me like a guy that is studying toxicology or something. I'm seeing me like a Lego builder. So I just constructing peptides. And when Mariana gave me this invitation, and talk, oh, why you don't talk about the wasp peptides and other things? I was just thinking, okay. And then I discovered that we have done a lot in the mastoparans. And I will start my talk talking that for the beginning, I don't want to work with mastoparans. I remember that I have one student, a PhD student, a very good one, that the name is Osmar, and he just asked me to work with mastoparans. I say, no way, I don't want to work with venoms and toxins because probably we'll never sell that. I'm always trying to construct a product for animal health or human health or for cosmetics or something. And in my mind, I was just thinking, could be very hard to sell a cream with a toxin of a snake or something. But then this was not true. I will show you that basically the people do not make this connection like a marketing place. And I remember that this guy, I say, no, we don't want to work with these mastoparans. And then he lies to me. He say, okay, Octávio, no problem. And then he start to work in my lab with the mastoparans. And since I have a big lab, sometimes I lost the control of my students. And one day he just asked me and say, okay, Octávio, I need to show something. And I say, okay. And then he showed me very, very good peptides. And I see the sequences, and I do not found the sequences in my mind. So I do not remember this kind of sequence. And then he told me, okay, these are toxins from wasps. And I say, what? And the guy say, take it easy, calm down, don't be rage. I will show very nice results. And when he showed me all these nice results, I say, okay, Osmar, I love you more. And now I love you also the mastoparans. So was interesting because in the beginning I don't want, and now my three main products are coming from toxins, and this is for me is quite surprising. So today I'm just coordinating three institutes: the SC Nova Biotech in Campo Grande, that is involved in bio-inspired products for animal health, and now agriculture because the government asked me to focus also in pathogens of plants. The Centro de Análises Proteômicas e Bioquímicas, that is my old center, now 25 years. It's a lot of time, I'm getting old. And the National Institute of Science and Technology for Bio-Inspired Products, that this year make 10 years. And today I will talk a little bit about the mastoparans. So, I think that this is... Okay, normally we start with natural products. I always, like most of people here in Brazil, started purifying peptides. I never did this before with wasp venoms. And then we work with clavanins, with Centridium muricatus, that is CMP5, and the plant peptide. So we just find a lot. And then this lead me to a problem, or many problems. We can publish a lot with these peptides, but we never go to the market. It was also a big space between the product development and the paper. Why? Because we have a lot of complexity, low activity, high and specific activity, aggregation, difficult to purifying, so many problems that now we develop some platforms to transform these peptides in something that could be useful for the industry. So mastoparans are targets for peptide design, so we can use these mastoparans as the base of new products. So a very nice charge distribution, a fragment excision, so we can cut this peptide and use part of them. I can apply D enantiomers in order to avoid the proteinase destruction. We can use alanine scans. We can use deamidation to protect them. And then they have multiple targets. So we can also find some mastoparans that destroy cancer cells, that could be very to erythrocytes, to fungi, and also to kill gram-positive and gram-negative bacteria. So once we have been using these tools to construct new peptides, we started from the beginning. So my first mastoparan that we published was a derivative of the mastoparanel. So mastoparanel is a very short peptide, and then one thing that I like it most is to study the distribution of the peptides and the amino acids inside of the peptide. How I said, I think in the peptides like a Lego. So I'm just thinking in the amino acids like blocks. My father was the first distributor of the Lego in Brazil. So I was the kid that was just putting the Legos and the pieces here in Brazil and trying all of them. So when I was very young, I do not have other toys, so I have only Legos. And my father is the guy that was thinking, okay, if you have a Lego, you'll be smart. So I have Legos all the time. So in the beginning it was very funny. In the middle was not so funny because I want a video game. And maybe I spend a lot of time with Legos. And then when I look to a peptide, I'm looking now like a pieces, and the amino acids could be distributed. And I always thinking that was very interesting because I have 20 pieces and I can construct many different things. So with a Lego, I can construct like a house, a boat, a Batman or something. Here I can construct anti-cancers, antifungals, antibacterials, and it was very, very interesting to understand how could I improve the activity against bacteria. So one thing that we observed that we need to change the hydrophobic moment and also the cationic position. So one thing that we done in the beginning, I don't know how to, no, how could I put a, no. Arrasta para o lado? Arrasta para cima. Ah, cá está. Ah, cara. Tem que fazer um negocinho assim. Vocês são chiques demais. So, we just added an isoleucine 5, and we switched it by alanine, and we added an arginine, arginine number 8, and for this thing, we just named it E5R8. So I improved the charge in the surface and thinking that we can just also improve the attachment in the surface of the membrane. And this was true, so we ran some molecular modeling, observed that I5R8 was much more attached to the surface of the membrane. And we observed that the MICs was decreased. So we really just changing 2 small pieces, and then we reach to an improvement of the activity. And this was also observed in the time QSIs, in which you observed that I5R8 was more faster to kill. Another thing is that we observed that the peptide keeps the mechanism of action in depolarization of the membrane. So we just compared the melitine, and we observed that we observe a similar thing that Mateus just observed, that we have like a detergent effect in destroying the surface of the membrane and allowing the changing of cations inside. And here we observe the effect of the peptide in a microscope, electron microscopy, and for 2 different bacteria and for candida. So we basically observed that the peptides go to the surface of the membrane and cause a terrible disruption. So we can just go further. And in this case, we just change a little bit inside. Another strategy was to add a tag at the end of our peptide. So we just have these mastoparans, and then at this point I was interested to help in the sepsis model. And then we are also interested to reduce the inflammation. So we just make a huge alignment of many and many other peptides. And then we discovered that we have a combination of 5 amino acids that we try to put at the end of the mastoparans to decrease the inflammation. So what we done? We observed that we have a conservation of this FLPAA in many immunomodulatory peptides, and then we add basically to the mastoparans. And then in the beginning, no, sorry. Como voltamos, nunca mais. Okay. We just, first we try with clavanins, that I do not show the data here, and nothing happens. And then we try with mastoparans, and then we observe that we can change the activity. And then at this point, I was working with Cesar, and he called me and said, why you don't put your tag also in brevinins and temporins and see what happens? And then we inserted these peptides in many, many different other peptides to see the effect. So we basically have a toxic mastoparanel, and then we add this tag, FLPAA, that is founded in the data bank, and then we created the mastmol. The mastmol was used many, many times in many, many articles, and this was the salvation of my student, because Osmar created this tag that works with the mastoparan. So we make a lot of assays, and we observed that this mastoparan MO was very, very effective against gram-positive and gram-negative bacteria. And then we observed that in the time kill that it was very, very efficient by fluorescence assays. And then we just run a lot of immune response tests, and we observed that this peptide, when received this tag, decreased the interleucin 6, the TNF-alpha, and reduced the inflammation associated with the antimicrobial activity. So at this point, we discovered, okay, we can add an extra block. And when we add this extra block, we have extra function. So that's cool, because now I know that a multifunctional peptide like mastoparan could be modulated, and I could add some extra activities. And then we just make a permutation. What's a permutation? My question was, if you have the FULP, this tag, in a certain sequence, and if I change the position, if I put the pieces in different places and combine with the different peptides again, so we perform these permutations with all of them. At the end, we have 500 different peptides, and we tested all of them and discovered that at the end, we need the sequence in the right place. Sometimes I can change one on another, but it was not like a question of, oh, very hydrophobic position. We need really the phenylalanine, the isoleucines, and teprolines in the right place to obtain the immune response effect. And then we run a lot of animal tests and observe that we can control the sepsis until certain point. So we can control until 80% with a 16 micromolar of activity. And then we tested several of them, the toppest one, and once more, the mastoparan MO was the best one. It was incredible, because my student created from the beginning, we make a lot of combinations, we just change the tag for other ones, and nothing was so good like the mastoparan MO. So he was forgiven. I like him very much. He expended a lot of money of the laboratory, but it was a good, very good paper. And then we ran a lot of NMR structures in order to understand how this tag changing the structure of the mastoparans. And you observed that when you have this peptide in the mastoparans, we have a different combination that calls a kink. And this kink, it seems that binding to something that I don't know what is it, and then perform the immune response activity. And then, okay, we just make this huge experiment with a lot of peptides, and I was just thinking, okay, we can create more. So basically, we just put a tag, we just change one on another, and then we created a mixture of peptides, 500. but maybe I could be more smart. Maybe I can use some algorithm to improve my hits, because it's quite expensive to expand 500 peptides to hit 1 or 2 or 3. Maybe I can use some strategy. So we use this Joker algorithm in order to find in all mastoparans and other classes of antimicrobial peptides some identity components. Oh, let's see. I'm so terrible with this. So with the identity components, so there are some amino acids that normally are found in these antimicrobial mastoparans, and there are some ambiguous components that change the classes, for example, a lysine by an arginine, or a phenylalanine by a tryptophan or something, and then are the wildcards. So the wildcards was used to create something that the nature did not offer me until now. So I add a piece over there that I cannot find in any place, and then we just add these fragments to scaffolds, to structural scaffolds that we can make more easy for the industry. For example, in this case, I was interested to helix. Most of people are interested in helix, including David Baker, that's creating the AlphaFold, because they are easy to predict. They normally are not very much aggregative. So quite nice. And then we just make a slide window, and then we create a lot of peptides. In this case, we created in the beginning almost 300, and then almost 120 different mastoparans. And I will not show all the mastoparans, but we have the R1 and R4 that was very potent. So was in the beginning I changed one on another, then I add a tag. Now we must use the strategy to improve the hits, and then we created these two different peptides. In this case, I have the same of you in TFA. We observed a helix, but when you add this in water, nothing happens. I have a lot of difficulty to see the NMR because there is a coil, and you cannot predict the coil. It's like a crazy peptide. And so we have a very potent peptide, but the next question is, if I put salt, or if I have temperature, do I have any problem? Because this is one of the limitations of the industry. See, if I have salt and block it, the positive and the cationic amino acids, so I have a problem, because in plasma I have some salt. Normally in the creams and other things, I have salt also. So we just tested against multiple concentrations and multiple in different salts, and we observed that these peptides do not have the salt not have any effect over these peptides. The same was in the temperature. We can keep this peptide active until 50 degrees Celsius, what is quite okay to go to the field. And then we just run some SPRs in Brisbane with Angeline, that is a suite of person, and then we observed that this peptides was very affinitive with the membrane. So we just created something, in this case, no, in the, oh, okay. In C, we have mastoparanel, and the R1 and R4, we have two different peptides generated from the mastoparanel that show more affinity to the lipids. What leads us to better understand the mechanism of action. So once more, we just run some molecular modeling experiments, and now we are just trying to simulate the peptides with the membranes, and also a different distance, because when I give the peptide was not just inserted in the membrane. We are trying to simulate what occurs when I have the peptide floating and then flow to the surface of the membrane and go inside. And what we observed, when you have these modifications, we have a more fast affinity of the peptide in the surface, and also the movimentation inserted inside of the membrane. Now, one of the problems that we need to solve is because we need more computational power to have thousands of peptides over a huge membrane and see what happens, because it's always difficult for me to believe in that one peptide will do a lot of destruction in the membrane. Well, we tested other activities of mastoparans. So we tested also antiviral activities. We see that part of the mastoparans included the I5R8 and mastoparan modified the mastemo, impede the adsorption and penetration of herpes virus. We tested also in this big project with Caesar in the coronavirus. So we just tested, and here, when you have this not so blue circles, it showed me that the peptides avoid the destruction of cells, of liver cells, and the mastoparan MO protected almost all of them in different concentrations. Here we tested a lot of other peptides that are not so effective. In the case of mastoparan L, we don't see this protection, but when you see the MAST-MO, we can see the protection against virus. And then we just start this activity in order to try to understand how this peptide becomes also antiviral. It seems that this peptide bindings to the lipids that comes from the vesicles that the virus carried, but we do not find any evidence that this peptide binding directly to the virus, maybe to the membrane, but not to the virus. And then we have a lot of studies with anti-cancer tests, and then we show that MAST-MO, MAST-L, and other derivatives were capable to control the tumors. In here, we just combined with gemcitabine and mastoparan, showing that we basically reduce the tumors a lot. So then we decided to go to a place that I could generate a product. So in the case of cancer, it's almost impossible, even because now I'm inserted in the pharmaceutical industry. I'm just coordinating the development strategy of MS in Brazil today, but they are not interested in cancer or infections. Basically, they are interested in other kind of like immune responsives and other things. So I was just thinking, okay, I have a lot of peptides, and normally they are much more interesting to peptides that they develop by themselves, not by buying for anyone from the university. And then I just focus in animal health. So I'm thinking, okay, I can contribute by improving the quality of the foods in our country. So if I have some peptides that decrease the bacterial activity, and then we do not have any residue, we just can use peptides with a green technology that avoid the contamination of milk, meat, and other things. So we just use the mastoparan-like peptides that we already created to test it against the Staphylococcus aureus that was very, very terrible for bovine mastitis. So in this case, we tested once more the MO, the R1, and the I5R8. So we just observed that these peptides were very, very effective against the Staphylococcus aureus that comes from the milk farms. And then we just collected Staphylococcus in many parts of the country, in Minas Gerais, in Mato Grosso do Sul, and in other, because we need to challenge our peptides with the worst that we have for the producers. And then we have some candidates. And then we just observed this effect once more, the helix and blah, blah, blah. And here are the three worst. The Staphylococcus aurora that destroyed part of the milk was basically destroyed by I5R8. Mastoparan MO is more or less, and R1 was not so good. So in this case, we just focus on I5R8, and then we observe that it's not effective against cow cells, and then Aurora versus the I5R8, we have an effect in 16 minutes. So in 1 hour, we just make an injections in the uber of the cow, and then we can see that. And then we observe that this peptide could be stable in milk. So one thing that is very, very important is if I use the peptide, it not be destroyed in the place that I'm using. So we just put in the milk. So we have 100% of the peptides with 0 hours, and after 6 hours, we have 20%. After 12 hours, we don't have any residue. So we have the effect of this peptide for 6, maybe 10 hours, and then we cannot find this peptide anymore inside of the milk. What reduce the levels of the toxins and the residues inside of the milk. So in this case, the industry was very, very happy because, okay, it is very, very effective, and then I do not have any residue. And then we just run a lot of fluorescence microscopy and observed by living dead cells that at 8 micromolar we can destroy, including the biofilms of the Staphylococcus. And then this is the Staphylococcus aurora. This is very terrible. When I talk about the producers with this stuff, they say, oh, God. And then they give me this. And you observe that they agglutinated all the time. They are a little bit different of other Staphylococcus aureus. And here is atomic force microscopy. When we observe that in the left side of you, we have the staph. And when we treat it with the peptide, we have the dissolution and dissolve of the bacteria. So we just observed that the peptide also decrease the size of the staph. When you add it, we have a destruction and a reduction of the staph size. And you observe that at the same time, when you have some candidas that we remove it from the Uber, we have the same effect. And how it works? So we just run some experiments with cryo-EM, and observe that the surface of the peptide binding to the surface of the membrane, and then cause a curvature that makes something like this. So when you see this like a detergent, we observe that, we can see the holes. But at the end, it's not a very, very well organized hole. It's basically like remove a vesicle in the surface of the bacteria and creating these vesicles and destroying step by step the bacteria. So now, how much time do I have? I finished it? 15? Ah, tá bom. Okay, sorry. No, because I was not thinking about the time, so if you give me more time, I can talk all the day about the peptide. So, but it will not take so long. So now we are just taking this strategy for Pantanal products, and we included, because in the beginning it was not included wasps, but now we started to sequence genomes of wasps from Pantanal also, to find a new sequence that I cannot imagine or figure out, because I always thinking, okay, I can construct some Legos with the peptides, but nature have much more knowledge than I am. So sometimes I need to look to something that I cannot even think about it. So one of the strategies is sequences a lot of animals in Pantanal. So we just started to sequence including wasps, to see if I have differences that I cannot manage by using my bioinformatic tools. So we are just sequences, and for now we have almost 2,000 bioinspired products that was also involved in tests. One of them that is derivated also for a toxin is a anti-mastitis, like you said before. It's better than the mastoparan I5R8. And then we just have this molecular dynamics, and NMRs, and other things. And we just discovered that we name it now MastPep, was very, very potent against a pool of Staphylococcus aureus again. So we just improved it a little bit more with the strategy of bioinformatics, something that starts from a mastoparan. And then we tested against many, many different things, and also against cows, because at the end I don't want to kill my cow, I want a happy cow. And then we tested macrophages, mammalian glandular epithelial cells, and erythrocytes, and nothing happens in this pharmacological window. And then we run first a model with mastitis in mice. So normally it takes a lot to have the cows, so the Uber of cows is huge. And then I was just thinking, okay, could be to see how the peptide go inserted in the milk or in the Uber of the mice, and see what happens. And what we observed is with 64 micromolar, we have a good activity against staph, and it works. So basically, we have a treatment with a MastPep, and it works. At this point, then I go to Ouro Fino Saúde Animal, and then they was interested in have something with us because we have not only developed the peptide, we have understanding the mechanism of action, and now we have some in vivo activity. But then they asked me, okay, did you try with cows? And at this point, I do not have cows in my house. And then I always tell, we have impasse. We have a difficulty between me and the company, and then they say, okay, if you try with the cows, we can think in a contract. And I say, okay, how much is the guy say X, would be a good investment, and then I buy 10 cows. So I buy 10 cows and put in a farm of a friend of mine that works, and make an agreement with my friend. I say, okay, you take care of my cows, black, tiger, panther, and put names in all of them. So they are my friends now, very, very nice cows. And you can use the milk to produce cheese, and you pay for the meal of the cows, and I pay for the cows. And the guy say, may I do a barbecue? And I say, no, don't kill black tiger, no. No, never. So we make an experiment, a controlled experiment first, injected the Staphylococcus aureus in the cow. So I have people in my team that are very, very good experts in animal tests, and then we used a very low number of animals, and then I observed a very huge standard deviation. So we can see a possible effect at 96 hours, and at 120 hours is quite okay after several treatments. It takes me a lot of peptides. I expend almost 700 milligrams of peptides to do these small tests. But this was enough. When I showed to the company, the guy say, okay, it seems that it works, but now we need to improve the number, and now in the next year we go to a field test with 500 cows. So they offer to us the possibility to pay the rest of the experiment. Another thing is that we observed the milk stability. So one thing that I'm just selling them is that I cannot be so potent like a ciprofloxacin or other antibiotics that they have in the market. But I never leave residue. So this could be important for exportation of the milk, yogurts, and other things. And I reduce the possibility of resistance, because if I don't have residue, the pressure of the selection is lower. And the guy say, oh, that's cool, this is very nice, could be very, very important for Europe. And then I think that's it. So after the treatment, after 6 hours, I cannot see any residue of the mastpep, what is very interesting for us. So it finished, not yet. We are just trying to sell our second product. The last one was a cosmetic, so that is not from our brand, and for this reason I do not talk about this. And it was very, very, a very good hype, because one of the persons that I was using our product, peptide base, was Katy Perry. So was very, very interesting, the effect it causes in the cell of the product. One day, Katy Perry just opened the purse in one talk show in the United States and say, oh, I like very much of this product that is for skincare. And then in the next day, they sell almost 600 flasks of the peptide. So it was something very amazing. So now I'm just thinking, if Katy Perry don't want to drink my milk with my peptide and say, okay, I love this milk, and then we can use more peptides to create some products. So now we are just thinking to improve a little bit more, because now is another hype is artificial intelligence. So what's artificial intelligence? It's just a different way to see the same that you are seeing. Now I'm a guy that is constructing Legos, but now I have a robot in my side that is telling me, don't do that. Okay, this you can do it. So I have an imaginary friend that probably I had when I have 10 years old that helped me to construct Legos. So so we created a platform to improve our peptides. So we're using a lot in order to avoid the errors and improve the hits. One important thing that I applied in this platform is because I always remember when my mother was hard with me. So when I do something wrong, I received a very hard penalty from my father and my mother. And I remember when my mother gave me a sandal in my bags and say, "Stop it, doctor, stop it." But I remember sometimes when my mother and my father say, "Good for you." So I apply the same in the platform. The negative one was much more powerful than the positive one. I give much more penalties when my platform commit a mistake than congratulations when the platform is improved. So we used Lime, that's a local interpretable model, a shape, and another IMP prediction algorithm that we developed with Caesar and other guys. And we add other kind of information. So at the beginning, I was just looking in pieces. So I was looking positive pieces, hydrophobic moment, distribution, but now I'm looking also to the target. So normally the peptides could do many different things. So it can create a hole in the membrane. It could bind in another thing like a protein or something. It could make a curvature in the membrane that could be negative and positive. So I give him to the system all this information. But because at the end, the artificial intelligence could provide me something good if I give a very, very huge data bank. So we expand last year just giving information about antimicrobial peptides, molecular mechanisms, the most important amino acids, and then we created this platform that we are testing now. And then we used these parameters like spontaneous curvatures, negative pressure, positive pressure, toroidal pores, the composition of bacterial cell, the capability to cell that, and then we used associated one thing that could make more easy, that explainable artificial intelligence. So when you put all this information, he tell me, okay, don't put this amino acid in this point. Don't put this one in this point. For example, in this case, the blues are the negatives and the reds are the positives. My team like to be inverse. Normally for me it's more intuitive to red be negative, but not in this case. So for example, aspartic acid, never use it for antimicrobial peptides. If you use it, you have a very low probability to hit or to create a very, very potent peptide. Otherwise, when you put the lysine, we have much more chance to hit to the goal. But you can say that in some cases, in very small cases, the aspartic acid could be there. So I have very small spots in red that show me, okay, don't use it in 99.9% of times, but sometimes you can use it. The lysine is the same. I can use it in 99%, but 1%. And then comes our brain, because at this point, it's not just to give to artificial intelligence and then receive it something and say, oh, thanks, artificial intelligence. If it was so easy, we can cure everything, but it's not so easy. It's much more complicated than I think. And then we created these new peptides, and now we are using something that we know, we named molecular reinforcement learning. So our prediction model, with all this information, create the peptides and classify the AMPs and non-AMPs. And then we have anti-new Klebsiella or something, and then we have a high reward sequences, and we have chemicals restrictions, and then we have a sequence generator that gives me the new sequence. And then I go to the bench and test and say, okay, now I have this, and these are okay, these others are terrible. And then we return the information and say, okay, thanks for this 4, I hate you for this 500, so we start again and start again. So we spend a lot of time just synthesizing, testing, and returning information, and synthesizing, testing, and returning information. For now, after 3 rounds of these strategies, we have 286 sequences. Among them, 4 are from mastoparans, and 1 is very, very similar to mastoparan MO. Once more, thanks, Osmar, for being a hard head for me. And we have now 15 new sequences that we are trying to prove that are very, very good. And this is the PepK10. It works very well. It's not like I like it, because I don't know if it's perfect. For now, nature is much better than me, but we are trying to improve it. So now we are very near to have a very, very good peptide. And then what's the new strategies for us? So first, better understand the molecular of mechanism of actions of of these antimicrobial peptides, because until now, I really believe that we don't know too much about the mechanism of actions. We know that destroy the membrane, we know, but we don't know almost nothing. Every time when you challenge this bacteria with the peptides, for example, in this case, we discover so many things when you have a resistance of these peptides. We make a very nice paper with proteomics, and we elucidated at least 20, 25 mechanisms of resistance with the peptide. So basically, the peptide is binding something and give me a lot of more information that I need to use it to create peptides that will avoid this resistance. So the new targets for me could be important. Another thing that we have discussing a lot with pharmaceutical companies is they don't like the idea to have a peptide that binds into a membrane. A membrane is not an easy target, so it's not a very, very replicable target. They want something very, very specific. For example, what lipids and what part of the lipids, the peptide binding, and how it works. So when I say, okay, I have this antimicrobial peptides, okay, but what is the target? And I say, could be membrane, could be a protein. And the guy say, okay, Octávio, very nice paper, I don't want it. So they are very restrictive with this. They like the idea to find something with different protein or something. And maybe it could be this. The new strategy could be the redesign the mastoparans to a certain different protein, and then created something that could block it with a very high specificity. So I think that is it. I always trying to show like a history, is because science is always part of my life, and peptides is part of my heart. So I like very much of this, and I like very much of my partners. I have a lot of partners in many parts of the world. And now we have a lot of funds, because this is quite expensive, how you know very well. So we have CNPq, CAPS, Fundec, that's the foundation of Mato Grosso do Sul. We have the cosmetics, IBA, that is providing us a lot of funds to discover new creams. FAPDF, the MS, the pharmaceutical one, FINEP, OneSkin, that we sell the first product with them. Peptides, that is one of my startup companies, and the Instituto Vita Nova, that is providing this immunomodulatory response. And these are, oh no, come on, my team. I need these guys. They are the responsible for everything. Yeah. Come on, no worries. Because it's a lot of work here, and these are the guys. So in the left side are my team in Campo Grande, and the right side is the team here in Brasilia. Now it's a little bit higher. This year was very, very good. A lot of people just come. And I always thank you to these guys that are working every day in the lab and have all the frustrations to discover 499 peptides that do not work, and one that is working very well. This is the Insta and the TikTok and the part of distribution of science. I do not make short dances. I do not do this, but I tell a lot about how to be a scientist, or how could be important to be in love for the thing that we are doing all the days. Okay? So thanks once more, Wagner, for all the partnership. You are always very, very important for us with the part of mass spectrometry, Mariana, about the discussions about antimicrobial peptides. Thanks a lot, guys.
Okay, thank you very much, Professor Octávio, for the great talk. It's open to questions.
Thank you, Octávio. Great talk, as usual. By the way, I also love playing with Legos.
I can imagine. When I think in one person that likes Legos more than me, I think that is you.
Yeah, I had a great time in Denmark. So, I was thinking about one of the peptides, the tagged one, that has antimicrobial activity, has anti-inflammatory activity, but has some influence in sepsis, but not that much. I would expect more for that peptide. So I was thinking maybe this anti-inflammatory activity is interfering with the antimicrobial activity of the cells. Did you test something on phagocytes?
This is a very good question. In fact, when we just submitted for the first time, the referee just said that it was not a very good strategy, because when I reduce the inflammation, I probably reduce also the defense against the microorganisms. So I think that we do not test it, but I think that you are right. So we have this effect when you reduce the inflammation. Part of the animals basically die, not because we don't have a correct effect, but because he lost part of the defense, and then we do not have the protection that we expected. In fact, when I compare the tag with the peptides and the tag with the other peptides, I don't see an improvement very much with the peptides without a tag. So we have something like 10 or 12%. That was enough to convince the referees that it was a nice history, but was, for me, was not a really amazing improvement. I expected much more. Another thing that I do not show here is when I have only the tag, I probably need to have the effect. But the effect only the tag is very low. So I can see the pattern in the bioinformatic analysis. When I add to the peptide, I can see something improving, but alone is not working too very well. So at the end, I see an effect, but I don't know nothing about the mechanism of action of the response, of the immune response. What I have been learning this year is that when you have something very difficult with microbiology, is much worse than immunology. So immunology is I know that part of the people here could be from microbiology, but the immunology is terrible. You know, so many ways, so many things that could happen. So and it's very unpredictable. But yes, the question is very good. It's a question. Maybe we can test the macrophages and the leukocytes and see the migration. I think that we never tested this before.
We're doing that, so if you'd like to.
Yeah, for sure. Good. Okay.
Okay.
Oh, fantastic results, Octávio. I was just wondering about that peptide, mastoparans MO. Did you check something regarding the membrane insertion of the peptide N-terminus? Because probably that phenylalanine residue at the N-terminus has something to do with it. So perhaps checking some analogs and the effect of this phenylalanine on the membrane insertion could give some insight on the great results it has shown. Thank you.
Well, I never mark with the fluorescence this one to see if the insertion is fast or not. I know that we have something because we can see the curvature of the membrane, so probably it was inserted in a right way. But I don't know about the velocity, and because normally this phenylalanine could disturb everything. In the beginning, I was thinking, okay, this disturbance could be good because we destroy the organization of the lipids. But at the end, I think that could be hard to put the phenylalanine inserted inside of the lipids. But I never tested before the fluorescence. I think that is a good idea to check the evolution of the entrance of the peptide in the membrane.
Yeah, we could check that doing NMR as well. So yeah, we can have a chat afterwards.
Yeah, for sure. That's nice.
Okay, next question.
Congrats for your presentation. You commented at the end that there could be a lot of mechanism of action, and I believe so. But when you change your peptide sequence, you are focused on the pore formation only, or another mechanism? What is the logic of changing the peptides and the flag as well?
Okay, this is a very nice question, because in the beginning, I don't know. I just focus in kill bacteria. So I was not interested in the beginning to see in what is binding or not. And then I was focusing in have a surface of cationic amino acids, and then another one, a very hydrophobic one. So my idea in the beginning, as many others like Bob Hancock and Mike Zaslow, was put in the surface and then make a flip and go inserted of the membrane. But at this point, I believe that was not enough. So the choosing of distribution is okay, is important, but also if I change the position of the amino acids, I can be more effective against certain kinds of lipids. So, for example, if I look to the mammalian lipids and the bacterial lipids, now I can predict what kind of amino acids I can put in a certain place to have more affinity to this kind of one. But now I'm interested because with this paper that we published before, we just get maybe 12 proteins from Klebsiella that appear, and it looks like very important for resistance and for infectivity. So now we are trying to design peptides that bind into these specific proteins and see what happens. So some of them probably not happens nothing, and then the bacteria say, oh, because bacteria is very, very adaptive to even to the peptides. But now the choice is according to the target. In my mind, I don't know if it's the right strategy, but now I'm trying to be more specific. And the tags in the beginning was according to the data bank. So I found a pattern, and then I just follow the pattern that the nature gives to me. But now we are trying to do the same with immune response. If you took the papers of IDRs from Hancock's lab, we have a very nice patterns of immunomodulators that could decrease and improve the inflammation. So we can use these patterns, and also in the information about the interleucin receptors that they touched. So I saw that some of these peptides bind into EL6 receptor, and then we can choose the tags according to the binding to the surface and avoid the interleucin. So now I'm just thinking much more in this direction. I think that I spent 10 years just trying to learn with a lot of mistakes, like this was okay, this was not so okay, but now I'm trying to focus. But this is a good question also.
Okay, any more questions? Okay, thank you so much, Professor Octávio.
Thanks. Eu roubei uma água aqui, eu podia, né? Ah, tá bom. Saber, né? A gente já tá bebendo água. Água alheia, é. Deixa eu botar isso aqui.